was a messy division. Whose appointment's next? Well, soon-to-be cells. Shall I divide them by meiosis or mitosis? Mitosis is a type of cell division which results in new cells with genetic material identical to that of the original cell and each other, whereas meiosis is a type of cell division which occurs during the formation of gametes, haploid reproduction cells. I think mitosis is fitting since I want them both to be equal by the end. But either way, by the end of this they will have gone through the cell cycle, the repeating set of events in a cell's life.
I'm done of sharing life with him in this one cell.
She's scraping my style in our cell.
Okay. I don't see why you'd want to be doing this after you've been growing your assets for so long in interphase, but in order for you guys to separate, you're going to have to go through mitosis. It's a type of asexual reproduction, which is the production of offspring from one parent cell, like you guys, and I'm willing to help you with this.
Whatever it takes to last divide.
Well, it'll take 4 stages.
And what will these stages be?
First there's prophase, then metaphase, anaphase, and finally telophase.
That sounds like it'll take forever.
Don't worry, we'll have you separated in no time. Okay, so first in prophase, we're going to need to coil up your DNA into tight, compact, dense chromosomes in order to make this all easier to handle later on in the division. We'll also need to copy your chromosomes as well and connect these chromatids with centromeres. Next, we'll need to start breaking down the nucleolus and finally the nuclear membrane.
All that's happening is things being taken away or changed. Does anything show up here?
Well, 2 pairs of darker spots, what are called centrosomes, will show up. Are you guys a plant or animal cell?
Animal.
Okay, in that case, each of these will have small cylindrical bodies. These are called centrioles, and they'll be inside of the centrosomes. If you were a plant cell, you wouldn't have these. But in both cases, the centrosomes will move towards opposite ends of the cell.
And what happens next?
Don't be stupid. Next, spindle fibers made of microtubules radiate from these centrosomes to get ready for This is known as a mitotic spindle, and it helps to divide our chromatids equally between our soon-to-be 2 new cells later on in this division.
Whatever.
Okay, moving on to the second stage of mitosis.
Let me guess. He knows all about this one too.
Well, actually...
Let me do my job, please. In metaphase, kinetochore fibers, one type of spindle fibers, move your chromosomes to the center of the cell, and once they get there, they are held in place with kinetochore fibers. Polar fibers, the other type of spindle fibers, will run from centrosome to centrosome.
I call those.
I'll ignore that comment for now and move on to anaphase, the third step in this division. Here, you will need to be sure you're going to go through with the division, because here, the chromatids of each chromosome will separate, and their centromeres will now be considered individual chromosomes.
I can't wait for step 3. I don't want my chromatids anywhere near yours anymore.
Ditto.
Okay then, in telophase, step 4, the chromosomes will reach opposite ends of the cell, and the spindle fibers will disassemble.
They better go to me.
As I was saying, the spindle fibers will disassemble, and the chromosomes will return to a less coiled chromatid state. A nuclear envelope will form around each of the chromosome sets, and a new nucleolus will be formed in each of your soon-to-be cells.
Well...
Yes, you are almost done with him at this stage. Next, during telophase, cytokinesis will occur. Since you two are an animal cell, as I believe you said before, cytokinesis will begin with the pinching inward of the cell membrane halfway, don't worry, Mrs. Eukaryotic cell, between the poles of your cells. Here, where the cell pinches in and separates, is known as the cleavage furrow. With help from the action of microfilaments, you guys will separate into 2 brand new offspring cells of about the same size.
Just out of curiosity, what would happen if we were plant cells?
Who cares? That's
Be nice, but good question. In plant cells, vesicles from the Golgi apparatus join together at the midline of the dividing cell to form a cell plate. This will later become a cell wall and will divide the cell.
Wait, whatever. Who cares? I have a better question. Can we split the cell plasm and organelle 60-40?
No, I'm sorry, but by cell division law, you'll both get about half of each of those.
Oh.
Now you're sure you want to go through with this?
As soon as possible.
Yes.
Alright, let's go then.
Wait one sec. What about cell division prokaryotes? Just curious.
Why does it matter? Who cares? Like, we are just
nice.
going to be divided.
Be nice, be nice. Prokaryotes have cell walls and lack nuclei and membrane-bound organelles. Also, the prokaryote's single DNA molecule is not coiled around proteins like it is in eukaryotes, so therefore it doesn't form chromosomes, but it is instead just attached to the inner surface of the plasma membrane, kind of the way a rope is attached to the inside of a tent. Binary fission is the process that prokaryotes go through to divide. Their DNA is copied, resulting in 2 identical chromosomes attached to the inside of the prokaryote's inner cell membrane. This grows, and eventually a new cell membrane develops between the 2 DNA copies.
Good to know.
Okay, let's go.
Remember,
Yes.
you guys are going to have to pass through 3 major checkpoints. First, the G1 or cell growth checkpoint. Here, proteins will decide you guys are ready to divide. The G2 or DNA synthesis checkpoint, where DNA repair enzymes check the results of DNA replication. And finally, the mitosis checkpoint. If you're able to pass through here, you'll be able to exit cell division and finish mitosis. But if these checkpoints don't work, you may go into a stage of uncontrolled cell division. Cancer.